Just want to thank you for the opportunity to tell you about SOR102. So it's a novel, orally administered anti-TNF 23 domain antibody for the treatment of ulcerative colitis. And just to quickly recap what ulcerative colitis is, it's one of the two diseases that make up inflammatory bowel disease, characterized by an inflammation of the lining of the gut. And in UC, that's constrained to the colon, and also it's a continuous disease. Now, patients with IBD report preferring oral tablets to other delivery modes, and that opens up two routes of delivery. So one are absorbed for systemic exposure, and the other, which I'm going to talk more about today, are delivered locally for direct action in the gut. So this offers up the opportunity of potential safety gains because there's minimal systemic exposure and less systemic immune suppression. But it's not easy to get protein drugs to the gut, not least because of all the proteases which are in the GI compartments on the way, and that includes inflammatory proteins of the lining of the gut. Also, intestinal transit times can be long, up to 45 hours in the most severe UC patients. So what you need is really robust drugs to achieve this. And there have been a number of modalities that have achieved this to date, and I'm going to go sort of left to right for these. On the left, you can see small cyclic peptides that are stable to the gut, like icotrokinra and PL8177. And on the far right-hand side, you can see larger classical antibodies, up to 150 kilodaltons in size. And a good example of these is Avaxia's bovine polyclonal anti-TNF that actually showed some efficacy in UC, but eventually was not continued. And just to note as well, there are a number of attempts to either protect or inject conventional approved antibodies for systemic delivery. But I'm going to focus today on VHHs, single domain antibodies. And you'll be aware already that these are the variable domains of heavy chain only antibodies. And there now exist a number of good examples of these that have been stabilized for delivery direct to the gut. So from our own pipeline, we've got V565, SOR101, which is an anti-TNF. But recently, Genentech reported a stabilized IL-23 receptor clone, and Novamab have also reported an orally delivered TL1A. And so why do VHHs have an advantage over conventional antibodies? Well, conventional antibodies are large, they're expensive to produce, and they're difficult to formulate into the concentrated formulations required for oral delivery. They're also not traditionally stable to the challenges of the gut, particularly in the lining of the gut. There are metalloproteinases which can clip off the Fc domain. So our solution is to use humanized single domain antibodies. And these have a number of properties that make them good building blocks to design orally delivered drugs. So robust production, stability in a range of conditions, and also the ability to link together multiple domains. So on the left-hand side of this slide, you can see our discovery process. And I've shown TNF and 23 here, but you can do this with multiple targets. And we start with a very sort of simple classical llama immunization, phage library panning to isolate clones. But then at the point where we start to look for potency and specificity, we also conduct an early stability screen in protease mixes. And what this does is enable us to pick out clones which have some intrinsic resistance to proteases. And then from there, we build further on that. So what we do is we protease stabilize them, humanize them, and increase production levels. And for the protease stability, we're doing things like changing out labile residues in the CDRs for more stable ones, increasing the overall compactness of the molecule. And also in the case of our anti-IL-23 arm, we actually changed out an entire framework, grafting in one from a more stable clone. And what we end up with is stable and potent clones that we can link together. And I've just shown SOR102 there is a bispecific at the bottom. And so there's good evidence, I mentioned, that these stabilized clones can now be delivered direct to the gut. And in both of the examples that I'm showing here, you can see in the top right hand of the slide, they use a mini-tab formulation. So these have got an enteric coat on them. And what that means is they're protected in the stomach, but when they move out into the small intestine, the pH goes up and the drug payload is released. And on the left-hand side of the slide, what you can see is a recent study from Ota et al., so it's from Genentech. And what they did was they developed a protease-stable anti-IL-23 receptor clone and dosed that orally to healthy cyno monkeys. And what you can see is there's staining in the ascending colon and also right into the lamina propria and submucosa, and also some systemic exposure as well. So orally delivered VHHs are able to get into the lining of the gut. And from our own programs on the right, you can see data for SOR101, which is an anti-TNF. And this is from a study that we conducted. It was a 7-day oral dosing study in UC patients. So we collected biopsies and then we did immunofluorescent staining. And what you can see just about hopefully is that throughout the tissue, you see it's sort of bathed in red, and that's staining of SOR101. But also there are these white foci points where you can see co-localization with CD14 or macrophages. They're expressing the membrane-bound version of TNF-alpha. So what we can see there is after oral dosing, target engagement in GI tissue. And so why is it important to be able to deliver drugs orally, protein drugs in IBD? Well, to answer that, we need to look at the evolution of advanced therapeutics. So there are systemic mAbs which are effective, particularly anti-TNFs. However, around a third of patients are primary non-responders, and up to 50% go on to be secondary non-responders. There are alternatives available, so anti-IL-23s are effective, including in individuals who don't respond to TNF therapy. However, injectable monotherapies are still limited in their use at the moment, and the market has trended towards oral systemic molecules. So there are currently approved options for this, for small molecules, so JAKs and S1P inhibitors, and these work well but have undesirable safety profiles. So there's also a need to increase and break the ceiling that we've seen with remission. And to do this, people have been exploring systemic mAb combinations. Most notably and recently, Janssen in their Vega trial tried a combination of a systemic anti-TNF and IL-23 that showed greater improvements in a range of clinical outcomes compared to either arm alone. So they really did break the treatment ceiling, but the safety of these long-term systemic therapies combined together is unknown. So our solution is an orally delivered dual TNF-23 domain antibody that acts locally in the gut with minimal systemic exposure. And that's SOR102 that I'm showing you in the top left-hand corner of this slide. So what we've done is to take that stabilized anti-TNF, couple it to a stabilized anti-IL-23, and that's produced as a single molecule in manufacturing and delivery, and that acts potently as a full-length molecule, so it inhibits and neutralizes both targets. But we've also introduced a lysine residue in the middle, which is cleavable by trypsin. So when it encounters that in the intestine, the molecule cleaves in two, releasing both of the stable arms to act independently of each other. We produce this at large scale in Pichia. We can purify it directly from the supernatant, and it's amenable to spray drying. And what we then do is formulate it into those enteric-coated mini tablets that I mentioned, and that goes inside an outer capsule. And that whole formulation is stable at room temperature for long term, so it removes cold chain requirements. When that's swallowed, the outer capsule almost immediately dissolves in the stomach, but the enteric coat protects against the low pH environment, but they're then free to move out through the pyloric sphincter, and as the pH goes up, the coat comes off, releasing the payload, and when it encounters trypsin, the linker's cleaved in two, releasing the monomers. And this slide just shows what I talked about on the previous slide, which is that both the IL-23 and TNF arm have comparable neutralization potency to already approved mAbs, and that formatting into and release from SOR102 retains that potency. In the intro, I mentioned that you need to be stable through all the GI compartments, and I'll go over this slide very quickly, but this just shows that both arms of that SOR102 molecule are stable to the matrices present in small intestine, large intestine, and also enzymes that are present in the lining of the inflamed gut. And so all of this gave us good confidence going into our phase 1 trial. And this is the format of that trial. So we did a pretty standard part 1 and 2 single ascending dose and multiple dose in healthy volunteers. And part 3 that I'll talk about in more detail was in mild to severe UC patients. As you can see in part 1, what we did was 3 ascending doses up to 3.6 grams, so a relatively high dose. And we saw no serious adverse events and no subjects discontinued. Likewise, in part 2, what we did was dose for 7 days, twice a day at a fixed dose, saw no adverse events. We didn't see any quantifiable serum or urine concentrations of one or two in its monomers, which you might expect because the gut lining is intact in healthy volunteers, and most of the serum samples are negative for ADA. So now I'm going to focus a bit more on part 3. So this was a signal-seeking study, and it was in, as I mentioned, mild to severe UC patients. The primary aim was safety and tolerability, but we also included secondary endpoints like PK and exploratory clinical and PD endpoints. We were targeting 18 patients, and they were randomized equally to 3 groups, so SOR102 twice a day, SOR102 once a day, or placebo. And we did this for 6 weeks. That's a relatively short study of this type, but at the time we initiated it, we were limited by the tox data that we had. And just to note that the single dose is 810 mg, so the BID group gets a double dose as well as being staggered in timing. And this is some of the data from that trial. So what you're looking at here is a clear and ELISA-based data. So this is actually cytokine engaging data of active drug in the stool from the single ascending dose phase. So just to orient you to what you're looking at, the black lines are the levels of SOR102, the bispecific, and the gold and the blue lines are the monomers, respectively, and they're shown on a log 10 graph. And so this is in the stool of individuals from the SAD phase. The cohorts are shown along the bottom, so increasing in dose along the x-axis. And because this was in the clinic, we were able to collect all stool samples. So what we did was dose healthy volunteers on day 1 and followed the drug in the stool over 4 days. And what you can clearly see straight away is there's a lot more monomer in there than there is of the full-length molecule. So this just validates the idea that it did get cleaved as it was going through the gut. And though there is some variability in the data, what you can see generally is that levels recovered in stool increase as we go from cohort 1 up to 3 and 4. 3 and 4 are actually very similar in the amounts that we recovered. And also, interestingly, you can see for the single doses that you start to see a plateau around day 2 or 3 that sometimes declines thereafter. So that's where we're getting sort of maximum exposure after a single dose. And stool recovery is important here because we don't see any systemic exposure. And so now looking at the multiple dose groups, what you can see again immediately is that the monomers again are a lot higher in concentration than the SOR102 bispecific, and again showing cleavage during transit. But with this multiple dose format, we very quickly have pretty high concentrations that are very stable throughout the course of the study. So they get up to a plateau at around day 2 or 3 and stay there to the end of dosing. And what is difficult to make out from looking at this, probably from where you can see, but the open circles and squares are the individual data points. And as I mentioned, we have every stool sample from this because it was done in the clinic. And from day 3 onwards, every single sample was positive for both monomers. So there's a real consistency there around delivering in this healthy volunteer study. And now I'm going to talk a little bit more about part 3, the phase 1b study. As I mentioned before, we were aiming to recruit individuals with mild to severe UC. Actually, in the end, most of the people we recruited had moderate to severe UC. And you can see it's a pretty visit-intensive study, but I just want to draw your attention to the left-hand side and right-hand side at the bottom. So we did sigmoidoscopies and collected biopsies before treatment and also at the end of 6 weeks treatment, so day 39 or 42. And that enabled us to do PD comparisons and PK comparisons at the start and the end, as well as looking at the lining of the gut by sigmoidoscopy. And just going to talk to you about the patient populations that I'm going to describe going forward. So there's 3 sets in general. One's a safety analysis set. So this includes all individuals who received at least 1 dose. And there are 2 efficacy sets that I'm going to refer to. One is the intention to treat set. So like the safety set, this is everyone who received at least 1 dose. But it also includes dropouts, so people who didn't get to the end of the study. And in this case, missing data is handled using the last observation carried forward. And in the absence of end of treatment biopsies, baseline values are used. So just note that that includes dropouts. And the other group is the per protocol or completer set. So these are all patients who completed the study. So we've got all the data from week 6, including those biopsies matched to week 1. And just to go over very quickly some of the UC endpoints I'm going to mention, Mayo score and modified Mayo score are some pretty standard measures that you'll see in various UC studies. Mayo score is made up of various subscores, including stool frequency, rectal bleeding, endoscopic appearance, and also physician's global assessment. And that physician's global assessment is left out of the modified Mayo score because it's somewhat subjective. And let's look at that clinical data. Okay, so this slide shows the clinical response by Mayo and modified Mayo score, and also symptom remission. And again, these are standard scores that you'll see in many contemporary UC trials. And what we can see immediately in the ITT population is that in blue, the BID group sees a 56 and 67% response in clinical response to Mayo and modified Mayo respectively, and a 56% increase in symptom remission. And that compares to 17, 33, and 0 for the placebo group. The BID group is somewhere in between, so we're seeing 43% in terms of Mayo and symptom remission, but the modified Mayo is very similar to the placebo group in that population. Moving to the per protocol population, what we can see is that in each of the scores measured, there's 100% response with the SOR102 BID group, but the 102 and QD and placebo are much like the ITT population. And just to highlight two key points from this slide. So the first is that the placebo rate is pretty low, which shows that these patients had active disease. And the second is that we're seeing a dose response. So the BID group, which is getting double and twice daily dosing, we're seeing more of a response in that group than in the QD. And this is just to show you some more data. This is clinical remission. So this is a really stringent endpoint. It's a gold standard endpoint as far as the FDA are concerned. And what we can see here is that by both Mayo and modified Mayo, so both of those are combined here, that only the SOR102 BID group is showing patients who meet this definition. And we see a 40% rate in the per protocol population. And this just shows some more data using a combined score of stool frequency and rectal bleeding combined. And just to note that you can see on the y-axis that the level here starts a little lower for the BID group, but there's a dramatic reduction down to day 42 in these symptoms. And actually, most of that has occurred by day 7. And moving even further, 80% has been reduced by day 14. So real clear symptom improvement within 2 weeks. But just to note again that the QD group and placebo group are not substantially different from one another. So now let's look at kind of PK in this patient population. So look at serum, immunogenicity, and fecal levels. And in terms of serum concentrations, we only measured one patient in the QD group that we saw active SOR102 IL-23, so the IL-23 arm. All other serum samples that we had were below LLOQ for all of our other assays, and that really confirms the hypothesis that at least at these levels of detection, there's minimal systemic exposure. In terms of immunogenicity, we did see 2 patients who had pre-existing ADAs, which is not uncommon for a biologic trial, and we had 6 patients who showed treatment-emergent ADAs, although the levels of titer were mostly below 240, and there was no apparent association between ADAs and efficacy or safety. So now looking at fecal concentrations, so this is very important in this UC patient population. And again, the graphs are laid out much like they were shown for the previous graph. So 102 in black, and then the gold shows the monomers, and the blue as well. And you can see on there that you've got dotted lines and straight lines, and those are the median and mean respectively. And you can see that they're actually quite closely grouped together. So just to note in this population that we weren't able to collect every sample, so we were sampling in the field. But what we can clearly see again is that the monomers are much more concentrated and are at very high concentrations in the stool compared to 102. So again, showing that it's cleaved during transit in the patient population. Also, they're very high and sustained both in the QD group and the BID group. But hopefully you can see that there's actually less variability in the BID group. So more stool samples are positive and there are higher concentrations throughout the BID group. And indeed, I think from day 3 onwards at least, every sample is positive there for both arms. So what we're showing there is a really high consistency in delivering both arms to the stool of UC patients. And of course, that's great, but you've got to get that into the tissue. And so we wanted to measure drug actually in the tissue itself. And so to do that, we used this novel targeted mass spectrometry approach that we developed with a group called Innovative in Nashville. And what this did was to look at 2 representative peptides, one from each arm. Just to note, this is not an antigen binding activity assay, but what it is very selective and very specific. And we were very pleased to see that in both the QD group and the BID group, in virtually all of the biopsies, we can recover monomer to both TNF and 23. One thing we also know is that while there is a difference in time between the last dose administered and the time that we took these biopsies, we also see that there's a difference in the level of monomers in the QD and BID group. So BID patients exhibited a 20 to 35-fold median level of the monomers. So it's possible that this is potentially driving those differences in clinical endpoint that we're seeing. Now, of course, getting it in the tissue is great, but you've got to engage target. And so we looked at 2 different PD endpoints to show that we were doing this. And one of these is shown on this slide. So this is looking at tissue cytokines by an MSD approach. And we did this in collaboration with the Tijtgat Institute in Amsterdam. And this is shown on a log 2-fold change from baseline. And as I said, this is an MSD format. And so what we showed was that we weren't able to detect either IL-22 or IL-23 protein levels in those biopsies. But what we did see is that, particularly in the BID group, there was a trend of reduction in cytokines compared to the placebo group for both general pro-inflammatory cytokines and also T-cell associated cytokines. And just to acknowledge at this point, it's a small data set. There's a lot of variability, but it was really nice to see sort of tissue PD results aligning at least at a group level with what we're seeing in terms of clinical endpoints. Just to know that obviously IL-10 you associate with being anti-inflammatory, and we see it go down in our biopsy study, but actually this is something that Janssen saw with Giselimab on IL-23 treatment, albeit their results were looking at serum levels of IL-10. So the second PD endpoint that we looked at was gene expression levels, and we did this using a gene correlation network analysis. So what we did was RNA sequencing, and we took that data and grouped it into what we call gene modules based on co-expression and functional connectivity using the BioNeuro-R package. Genes in a given module were compared to the GOBP database to determine the associated pathways, and also to a previously existing single-cell RNA-seq expression data set of colonic mucosal cells in UC. And the combination of these 2 analyses allows module to be given cell types or pathway annotations. And just to note, the limitation of this is that genes can only appear in one of the modules, and the name given to them is basically the dominant biology, but not necessarily the only biology in each module. But this data shows the results of that. So again, shown as log 2-fold change from baseline. And along the top, what you can see is 3 modules that we saw decrease, particularly in the BID dose group. And just to take you from left to right for those, on the top left, we can see alpha beta T cell module going down in response to BID SOR102 treatment. And this is a kind of biology that you might associate with IL-23. So indeed, TYK2, which is immediately downstream of IL-23 receptor, is in this module. Myeloid inflammation in the middle is reduced in both the QD and BID groups, and this is what you might more associate with innate biology, so TNF signaling. And on the far right, an interesting module that we looked at was B cells. So B cells, particularly naive B cells, are increased in UC tissues. And interestingly, this is becoming more associated with IL-23 response. So again, Janssen saw that when they dosed Giselimab, they saw a reduction in expression of B cell genes. So again, it's nice to see that these gene sets going down in response to SOR102. And on the bottom, we see sets of genes which you might more associate with tissue healing and repair. So we've got stem cell maintenance, transcriptional regulator MYC, and the unfolded protein response going up, particularly in the SOR102 BID group. So again, just to acknowledge that this is a small sample, there's a lot of variability in there, but it's nice to see a combination of innate and adaptive responses being reduced in response to particularly BID therapy. At the same time, we're seeing tissue healing processes improve. Now, while it's a small study, one of the benefits of that is that you can actually look at things by patient. And what we can see on this slide is a breakout by patient. So down the left-hand side, you can see all the patients, and each row is a different patient, and they're shown by group, so placebo, QD, and BID. And what we've done is to take on the far right-hand side of that slide is the data from the previous slide. We flipped it on its side, so horizontally, colored it red for bad and green for good. And then what we've done is compare that to a range of the clinical endpoints that we looked at, including ones that I didn't mention earlier today. And so each of those has got a different threshold, but basically it's red for no response or worsening for each of the endpoints, and green for an improvement. And what you can hopefully see with the BID group, so just in general at a group level, is that we are seeing some association between what we're seeing in terms of clinical endpoints and those tissue-driven PD endpoints. And so for the sake of time, I'm just going to focus on the last point here. So SOR102 shows a clear dose response in clinical and PD endpoints, and that's going to inform our phase 2 design. And that leaves me just to acknowledge the rest of the Ceriso team. So we're a small but very dedicated team. So those are all the names on the left, and also scientific advisors from our legacy group, VH², and also thanks to Matt and Ingrid, who performed the PD analysis, Alimentiv, who we partnered with to do part 3 in the precision medicine, and also just thanks to Magnify, who are the lab that host us at UCLA. And now just to mention that data, most of it has just been published in Lancet GI and Hepatology. And thanks for listening, and I'll take any questions.
Question. Yeah. Did any of the patients in these studies receive anti-TNF or anti-IL-23 prior? Has there been any analysis of that, or are they completely kind of naive in that sense?
Yeah, so we went for, I think I'm right in saying that we went for treatment naive. That doesn't preclude us from dosing those patients in future, but like I say, this was a really small signal-seeking study, so we basically set it up to kind of give us the best chance of seeing efficacy.
And then a second question, the mass spec approach for looking at PK in tissue, right? Any thoughts on using that and looking at, can you stack dose? Can you actually get a steady state? Could you, appreciating there might be technical hurdles to do that?
Yeah, I mean, the main one there is, that they obviously snapshots in biopsies, which we're really interested in. Now, of course, you might, take multiple samples, but in this study, there were a lot of visits, so we only were able to take at the start and end. But really what you'd like to see is a picture of how that looks over time, because all the other outcomes are the end of 6 weeks dosing, but that's just a moment in time.
It's just a moment in time.
And they're quite consistent between the groups based on that. But yeah, we would obviously like to do that, but it's a lot of sampling of patients to do that.
Thank you.
Nice talk and some interesting results from the clinical trials. I was wondering, the doses are quite high, twice daily. Can you speculate on why that is? Is it the target selection? Does it have to do with remaining stability, tissue penetration, or you need a systemic component? Yeah, so obviously overall dose is something we're going to look to tailor as we move into phase 2 and beyond. So this isn't necessarily the final dose, but we're not looking to thread the needle on dose. We've got good cost of goods. We can produce a lot of this stuff. And so basically we were looking for a signal in this study. So we're giving a lot, and it's not uncommon to give a lot of biologics. Something we are working on and have made good progress is actually reducing the number of pills that we're going to get to give that dose. But also, dose is something that may change, or total dose, as we move through clinical development.
Over here.
Go ahead.
So just a quick question, the lysine and the linker, do you really need it? Does it change the binding and neutralization of
I mean, the short answer is you don't absolutely need it. The full-length molecule does bind to both and will work in that format, but we just wanted to make sure that there was no kind of pulling of one target away from the other. It's basically just for efficiency and was a nice touch that we got to work. But it isn't required for any potency. It doesn't activate the drug. It's just to enable them to act independently, and so they're not bringing targets together.
Thanks.
So you sort of glossed over it, but you said you just eliminated a few residues to give it the resistance. Can you say a bit more about that? I mean, did you have to do any selections? How did you make it so that it stayed stable in the gut?
Yeah, so I won't go into too much detail on that, but there's some data out there we've released showing that some of them were just changing CDR residues, so lysines, we swapped to histidines. We've got a number of other changes that are more proprietary, but the one thing that really helped us out, particularly for the 23 arm, which I mentioned, was that one of the framework pieces of that just was sort of limiting stability there, and we weren't getting above a certain stability. But we've got other clones from our collection, so we swapped in a framework, and we were lucky enough that it grafted well, and that was what enabled us to make the IL-23 domain. But yeah, I can't tell you about every single mutation we use, but it's a combination of those things.
Okay. A couple of questions. When these therapies are traditionally given IV, there's normally, or sub-Q, there's normally an induction period prior to the maintenance dose, and it looks like you're more on the maintenance dose period. Did you consider that, or is it even possible with the oral delivery? And then also, is there any change in the serum level as the patients go into reduction or into remission?
Yeah, so obviously we haven't looked at remission yet, but we will do in our phase 2 trial. So that's going to be a pivotal trial. We're designing it so it meets FDA, EMA criteria, and it will have both an induction and a maintenance phase to check all of these. And we'll look at PK and PD there, but we haven't actually dosed maintenance. These were all individuals with either moderate to severe UC. And in these individuals, we're not seeing lots of systemic exposure, so we wouldn't expect that to go up for an individual in maintenance, but we do expect from what we know about the gut that there are endoscopically normal individuals in remission who have some disruption to the gut. So we still expect to get our drug there, and we expect it to be effective in both phases.
So do you feel that disruption of the gut is necessary?
To some extent, yes, but it doesn't necessarily need to be a full and active flare.
Okay. Any other questions? All right. With that, let's thank presenters again.